So my name is Kaitlin Sullivan, and I'm a PhD student in the Simbrowski lab. And today I'm going to be talking about some analysis packages that I created for mFISH during the past year as my time as a CONP scholar. So mFISH stands for multiplex in fluorescent in situ hybridization. It's a spatial transcriptomic technique that I helped get up and running in the lab. Basically allows us to fluorescently tag mRNA present in a tissue section with single molecule and single cell resolution. The way it works is that we hybridize 12 probes to a tissue section, amplify, and then visualize those probes using cleavable fluorophores, imaging 4 at a time. So after 3 rounds of imaging, we have a full 12 mRNA visualized within a single section. Then we can dehybridize all 12 of those probes, add another 12 for 24, 36, 48, et cetera, gene targets within a single tissue section. So one of the analysis packages that I've created is a Fiji drop-down menu that basically takes all of the individual rounds of imaging, registers them to the same coordinate plane, then segments the individual cell bodies, quantifies the gene expression within each of the targets that we've looked at, and finally creates a nice qualitative overlay image that we can look at. Beyond this qualitative image, we also have the gene expression matrices that come out from the quantification portion. For this, I've created a Shiny app. This is just a screenshot of kind of what it looks like. Basically, with this Shiny app, we can visualize the data, as well as use machine learning techniques like dimensionality reduction and clustering to further analyze the data. So here, using data from our recently published eLife paper in the basolateral amygdala, this is the overlay image that comes out from the Fiji app. And then using the gene expression matrices, we can upload them into the Shiny app, look at raw gene expression, both the spatial location as well as in these dimensionally reduced embeddings for raw gene expression, as well as hierarchical clustering, where you can simply take a slider, select the number of clusters you would like, look at them again in space, as well as dimensionally reduced embedding. And so now this technique is being used a lot more frequently within our lab. And so I've had lots of alpha testing that has gone on through my various lab members. In another amygdalar nuclei, Rennie Kendrick has looked at the central amygdala. So this is what comes out of our Fiji app. And then you can just put it into the Shiny app and do your various dimensionality reduction and clustering. Also, another recently published data set in the classroom done by Brianna Bristow. Here's what comes out of the Fiji drop-down menu. And then this is what happens when you run it through the Shiny app. And also recently, Dr. Larissa Krause has been using this for our human tissue experiments. So this is the human cortex using fresh tissue. And again, we can just run it through the app and quickly, without any coding, get all of your dimensionality reduction, clustering, and spatial information. And so hopefully we'll be moving on to sort of more beta testing soon, and this will all be available on GitHub as well as the Open Science Framework. And with that, I'd just like to thank my lab and the COMP for this opportunity. Thanks.